diseases. Continue.
Thank you very much. So, the title of my talk is Identification of Circulating Proteins as Biomarkers for Cardiovascular Diseases. I will not talk about many biology because I don't think anybody is from biotechnology here. Anybody is from biotechnology? Oh, so many. Good. Less engineers than biotechnologists. Okay. So you are MSc or BSc? BSc. So this is the area of research. I used to work in hypertrophy and also cardiovascular diseases. So we talk about these two. Cardiovascular diseases, the coronary artery disease. Coronary artery disease means that we do the atherosclerosis and heart attack. And the other one is the rheumatic heart disease. So we try to understand the first from the human sample, patient sample, what are the altered proteins before and after the disease onset. And also try to understand this in the animal model and the cell line model. Because in biology you cannot you have to understand the mechanism, then only you can establish the cause-effect relationship. To establish the cause-effect relationship, you cannot utilize human, you have to utilize the animal model, proper animal model. So this is an important area where one of the important diseases in India, the rheumatic heart disease. Because of the streptococcal throat infection in the early childhood, some of the people in the adult, maybe after 14 years of age, and 14 years age and onwards, their heart valve gets defective because the repeated streptococcal infection causes the development of antibody and it binds to the heart valve. There is a heart valve, there is a valve in there is mitral valve that actually connects the atrium and ventricle. That actually controls the blood flow between atrium and the ventricle. That is very critical. But this valve gets inflamed. Inflammation happens and thickening happens and fibrosis happens because of this infection, streptococcal infection. So that it looks like normal valve, and this is a section, the real human heart section, valve section. There are a lot of fibrosis, the angiogenesis, infiltration, inflammation, all this is established, and they control the normal heart valve, the postmortem sample. So what is the problem? Problem is how to detect the rheumatic heart disease. Rheumatic heart disease is detected now by echocardiography, imaging technique. But most of the people are actually, they belong from the lower socioeconomic strata, poor people. They are living in their unhygienic condition. That is why this disease is very popular, very common in India, Bangladesh, Sri Lanka, Brazil, all the developing countries, including Australia, because there are some aborigines. So before people when are coming to the doctors with the advanced stage, before the valve is get operated, they die. So the only therapy is to replace some penicillin therapy is there, but the corrective measure is to replace the valve with the prosthetic valve. You know the Suchitra valve is very famous, that actually lowered the price for the heart valve. So the people have to come there and then wait for the heart valve to get the valve, to arrange money, to get the hospital bed. Then a lot of time is wasted by the time they die. So the doctor thought that can we find out something in the blood which can detect and say that the rheumatic heart valve disease is there. Doctors can easily diagnose by seeing the echocardiography and also by symptoms, but confirmatory symptoms is not there. Therefore, we collected blood sample from the diseased patient, partly in the hospital in Calcutta. They are mainly in AIMS, PG, all right, government hospitals, large number of patients. And then we actually check the one protein level. That protein actually we hypothesized from our earlier research. So the protein level is PICB. What about how it is done? What is the protein of this? I'm not going to do this. This protein is actually protein which is required for the fibrosis. I'm shown in the histopathology that there is a huge fibrosis. The fibrosis is occurred because of the collagen synthesis. Collagen synthesis is necessary. Collagen is actually synthesized from a big chain. First the pre-collagen, and the pre-collagen is into collagen fiber. That is a fibrillary collagen. That collagen, during this processing the collagen, there are peptides are released into the bloodstream, both from the C terminus and from the N terminal domain. So we hypothesized that peptide can be measured in the blood. So we took the blood sample and measured. You can see there is a dramatic increase in the value of the peptide, several folds compared to the control. This is the control in the patient. Before operation, heart valve operation, after the valve operation. So after the valve operation, you can see the protein goes down. And now in this patient, the defective valve is not there, prosthetic valve is there. And we followed them up to 1 year, 6 months or 1 year, it comes to the normal level. That suggesting that the defective valve is the cause of this high level of PICP in the patient. And we did a lot of statistics to check whether this is suitable for diagnosis. So receiver operating characteristics, ROC curves, and compared to the other, we found the value which above and which you can diagnose as that this is the AISD and patent and published. And we also randomly check this level in other disease sample like leishmaniasis, other disease, cholera. It is not increased. It is only increase in the antibody disease. And then we try to establish some kind of the developing kit. That kit is this is the before COVID. So at the time, now everybody knows what is this lateral flow device, but earlier it was not known. So there is a device. So some band is there in the patient and not in the control. And also by ELISA, quantitative estimation, we found that this is very remarkably increased. We patented it. But that's it. After that, we actually start there because some level of signal is there in the control. Now this is the time for the intervention of the electronic engineering or the electrochemical, so that we can find out. Now this is outdated technique. People are actually designing smart technology. So now we are trying to collaborate. We have got the antibody also. We should need the antibody. Antibody also developed. We are trying to find out some company, not like this kind of kit, which is actually detect the very smart way, not by the traditional ELISA kit. ELISA will be outdated very soon. So that's the part one. The second is that is rheumatic heart disease. The second was the very common, most of the countries developing, reach for all countries, that is coronary artery disease. In the coronary artery disease, it actually related to cholesterol. Cholesterol is linked to coronary artery disease, which cholesterol decrease and expressed in the aorta, and that causes the plaque. And the plaques can rupture, so it can cause heart attack any moment. But cholesterol, actually, we have a very efficient mechanism of cholesterol synthesis. Because we need cholesterol in our body, in our cell, in our nerves, in our brain, from one head to tail, we need the cholesterol. So therefore, body has a very highly efficient cholesterol synthesis mechanism in the liver. Liver is the main storage cholesterol synthetic machinery, and cholesterol. So whether we eat or don't eat, cholesterol will be synthesized in our liver. Therefore, it is now proven that dietary cholesterol contributes 30% of blood cholesterol. So the cholesterol is actually synthesizing all the time from your liver. So that cholesterol goes to the peripheral circulation, but the excess cholesterol comes back to the liver via a mechanism for LDL receptor mediated. So LDL is a bad cholesterol, comes back to the liver. But there are other mechanisms. There is a basic mechanism of defense in the cell, in the body, that is the monocytes, macrophages. Macrophages are there around, moving around in the blood. Whenever there is something infection, macrophage will go and engulf it. It does same thing for the cholesterol also. It engulfs cholesterol and convert it into foam cell, macrophage, the developed macrophage, and that cholesterol is efflux out from the cell by HDL. That's why HDL is called, packaged into HDL. HDL is called good cholesterol. Then HDL goes back to the liver for breakdown. So HDL actually helps clearing your LDL, bad cholesterol. So this is the mechanism by which actually cholesterol is remain balanced. So one of our subject is this is called reverse cholesterol transport. We wanted to see reverse cholesterol transport and inflammation as a relationship. So most of the people living in the hall, even the younger people, are having some plaque in the blood, in the artery. It is a disease of the artery, not the heart. Arterial disease. In the artery, this plaque, everybody plaque is dissolved. So plaque is very small. It is inflammatory resolve. So few laboratories, including all have shown that if there is huge inflammation induced in the system, arterial inflammation, the whole plaque is going to the chronic inflammatory stage. And the cell, these macrophages that actually engulf cholesterol, remains there and become necrotic. It is a loose plaque. So this plaque can rupture any moment. So this actually, so therefore heart attack is not sudden. You will think that heart attack is sudden. Heart attack has some signal. So therefore, if you can find out the signal, then you can actually predict atherosclerosis. Also, at the present time, you cannot understand the atherosclerosis burden of the individual. So most of the people sitting here are plaque resolved. But if anybody has the unresolved plaque, it can rupture any moment. Most of the younger people have the resolved plaque. So the question is, can we predict the atherosclerotic burden of the individual? We can predict atherosclerotic burden. We cannot predict. We can measure by the angioplasty, angiography. But we will not go for angiography. The angiography is a very invasive technique. It can cause heart attack also. The angiography is done, angioplasty is done at the same time. If there is a problem, heart chest pain happen, then we will go to angiography. So there is one marker is there which can confirm the heart attack. When somebody had heart attack, goes to the hospital, and there is a test called troponin test, troponin to ensure the heart attack happens, not other pain. So, but there is no marker which can predict the heart attack. So our ambitious attempt was to find out the signature protein in the blood, linking cholesterol aortic inflammation, whether reverse cholesterol transport is critical for inflammation in the atherosclerotic inflammation, and we can have a clinical correlation with coronary artery disease. So we take the sample of the blood sample from the hospital, the very good hospitals, Apollo Hospital. So patient enter into the hospitals with the 6 hours of chest pain. The collect blood sample collected from the hospital where the patient and comes to the lab, and this is a very high resolution mass spectrometry. So by if you analyze the protein, it can identify the whole how many thousands of proteins in the blood. So basically, because of the inflammation is progressing, some molecules will be reflected in the blood. So blood is actually reflecting whole organ system, your condition of the pathophysiology, physiology, or normal physiology. Blood has everything. So if you can trap something from the blood, you can detect by non-invasively, without surgery, without angiography. So that is a purpose. That's why we took the blood sample, and it is easy to take the blood sample. So this is the case control design. So some cases, some control, and a lot of bioproteins, proteomics, just keeping. And then we try to understand the mechanism using the mice model, because we cannot do the surgery in human. So there is a mice model. This mice called the knockout mice. One gene is knocked out. So the mice has, if you feed the mice with high cholesterol diet, it develops the plaque. It's like human plaque. So this is the atherosclerotic plaque. This is a normal aorta. And also we have other mice. The PPR from knockout mice. The mice PPR for gene is knocked out because it has oxidation is less. So their produced oxidation is less. So the blood is always thick, white, high cholesterol in the blood. To correlate with the cholesterol story, we took this mice. And we took this sample, the data from the human patient and also control, like lipid profile, their cholesterol concentration, whether they are suffering from hypertension, whether they have blood sugar, whether they have smoking habits. These are the risk factors for atherosclerosis. So those we conducted and did the proteomics. We processed the sample and processed through mass spectrometry. So highly powerful mass spectrometry identified 7,000 proteins, 3,000 proteins. Some are upregulated, some are downregulated. And out of that, out of 3,000 proteins, we actually narrowed down into 6 proteins for further study. This is a panel, my clean panel for the prediction of heart attack. So these are SGLT1, SGLT1, PGLRP2, Phenylalanine, ABC5, ABC1. Some proteins are upregulated, some proteins are downregulated. So that if you design something with AI, it can normalize the minor changes. So there's some upregulated protein, some downregulated proteins. The protein level is increased in the blood at the time of heart attack, protein level is decreased in the blood at the time of heart attack. So let's see the story very briefly. SGLT1 is one protein which is released from the platelet surface. When there is a coronary occlusion or heart attack, there will be platelet will be activated, and from platelet surface, some proteins are released into the bloodstream. So that protein is called SGLT1. This protein has the relationship with the inflammation also. So the question is, if there is a highly inflamed plaque, the possibility of heart attack is very high. How to find out highly inflamed plaque? Well, possibly by using these markers. So you can see this number, amount. The amount of the protein is picogram, 460 picogram in the normal among us, ourselves. 460 or picogram, the normal people, normal individual, human individual. The protein level goes up to 2342 picogram per ml. So there is huge scope to make it a diagnostic marker because there is several-fold increase. It is not a minor increase, and it is an exact quantification by ELISA, enzyme linked immunohistochemistry assay. But most interesting thing is this group. This is the patient, heart attack happens, you know, angiography diagnosed heart attack in the hospital admitted, and this is a normal individual from the local population. But this is a group where they are suffering from they have some risk factors for atherosclerosis, either cholesterol level abnormal, or they are a smoker, there is hypertension. But they don't know, they know that some problem is there. They are called asymptomatic individual. See, the protein level is also reasonably high in these people. So they are the people you have to detect before that, so that we can predict, okay, it can have a heart attack in future. So, and then a lot of correlation with before a surgery, after surgery, not surgery, coronary angioplasty, before coronary is done, and a lot of statistical correlation. When the correlation with the allele, if the cholesterol is high, the protein level is high. So this is a very nice correlation with our sensitivity, specificity. We could predict the amount of protein above which it will be heart attack happen below it, and the relationship with this. And we showed that with the many other biological experiments, with mice experiment, with cell line experiment. We showed the mechanism. This protein actually helps binds to a protein, another protein, released from the blood, the platelet, binds to a protein, the macrophage cell surface, the macrophage cell, which is actually your guarding your system, taking cholesterol, removing cholesterol, and the cells are macrophage cell surface binding to this and making this protein, this pathway, activating this pathway, making the whole system inflamed. So this protein is high means it is not good. This is actually inducing the arterial layer into inflammatory state. So this is nicely published, this from the computational modeling and the journal. Second protein is also the ZZP1. This protein, so all the data is there. This protein is down-regulated. I say some protein is up-regulated, some protein down-regulated. This protein down-regulated significantly at the time of heart attack, and also heart attack with, and also the coronary artery disease, it also goes distally less, and also with type 2 diabetes. So type 2 diabetes has also cardiomyopathy, the protein level is very low. So this has a huge, in novel patient, novel subject, protein level is around 80 to 90, but it goes to say 40, 50 in case of CAD, and type 2 diabetes and CAD, coronary artery disease, is very less, 30 microgram per ml. This protein also involved with the whole atherosclerotic, this the inflammatory system. This protein, if it is balanced, low, it remains it drives it towards the inflammatory state. The protein is very high, then it actually maintains the inflammatory, it maintains the plaque, the resolved state. The inflammation is resolved. In this case, it was unresolved. This protein is also very important role in the inflammatory, arterial inflammation. Third one is mitronectin. This is the cell adhesion protein. All the data like this, all the data collected, and this protein actually goes down very significantly. This normal protein level is 100 nanogram, 90 nanogram per ml. Among our age, it will be 75. But in the normal students, the younger it is 100, 120. But in the patient, it is drastically reduced to 20 nanogram per ml, 20 microgram per ml. That is asymptomatic group, also reasonably less, very less. Not to this level, but it actually giving signal that these people may have heart attack within very soon, within 1 or 2 years. That is our hypothesis. But we cannot tell because it is not actually established as biomarker with the follow-up study. And we showed that we knocked down this protein also from the animal, and we showed that established that this protein is also involved in the inflammation of the plaque. The fourth protein is PGRP2. This protein is increased. So one protein increased, second two protein decreased, third protein decreased. This protein is increased, very high. You can see this is the quantity, 2,900 microgram per ml. Picogram per ml in the patient, in the normal individual. In the patient, in the heart attack patient, it is 5,300 microgram per ml. But in the asymptomatic group, it is 5,000, 4,400 picogram per ml. So there is a huge opportunity to play around this because there is a robust increase, and also the actual quantitative data, not the qualitative data. And also we can predict the occurrence of 7-second heart attack. There is a score called TIMI score. Using TIMI score, you can predict the occurrence of second heart attack. People who are having one heart attack, there is a possibility of second heart attack. So that also we predict, our marker can predict the possibility of second heart attack. And this is also the knockdown in the animal system and showed that this is actually involving the plaque rupture process. So this is published in a very elegant publication journal, that is Arthurs of the Clinical Journal. From the, so yes, the last the ABC1 ABC2 ratio. So one protein in the mass spec analysis we found that the which protein ABC1 is a cholesterol transporter. When there is a cholesterol, it helps the transport of cholesterol from the blood to the liver. If it is good, then actually cholesterol not recycled, broken down in the liver. Your cholesterol level is maintained. ABC1 is actually downregulated at the time of heart attack. We found that by mass spec analysis. We validated it by ELISA method. We showed that it is also decreased at the time of heart attack. But we found that what are the other similar proteins? And we found another protein, ABC5, that is also cholesterol transporter, but it is not known as a cholesterol transporter. That is increased. So then with many other experiments, hundreds of experiments, we established, and was a human experiment, we established this protein, this is a new channel, the new transport system we discovered. The already known transport system, this is the simulation data, computer simulation data, also wet lab data. This protein is actually is a transformant protein. It takes cholesterol from the macrophage and going out of the cell in the bloodstream. So this is called active transport. This is transporting cholesterol against the concentration gradient. It is known, and what we have shown that this is also cholesterol transporter capacity, the activity, the property. But the transformant domain is very stubborn. It doesn't open. It opens only when there is a high cholesterol. So we have discovered a new mechanism that yes, when there is a high cholesterol, another supporting system is there. When this also fails, we have shown that plaque ruptures happen in the animal model. So with this, all these also very published in good journal. And so this is technical story. I will not go to it. So in a nutshell, actually we have established a 6-panel marker. They all are published in a very good journal. From human sample, established in animal model and cell culture. Now these proteins, actually we have some, we cannot claim as a marker. Some of the proteins we have really found that it is decreased or increased, and somebody has some chest pain and angiography done from our cohort. But we cannot tell them unless we have a follow-up study. So this is the difference in biology and the electronics. You don't have to have FDA permission, you don't have to have other experiments. Here you have to show 5 years follow-up study that if my proteins are altered, heart attack happening. Or among the heart attack sample, my proteins are already altered. So this is called prospective study, follow-up study, or the retrospective study. Already heart attack happens, hundreds and thousands of samples you have to collect and you have to show that yes, my protein are in down regulation, or the panel of 6 protein, their heart attack happens. Then only you can predict the heart attack. So the panel is associated with disease risk and disease severity in coronary disease. Specific cutoff values are obtained, mechanism is also obtained. So the future direction is a longitudinal validation called follow-up study. What CSI, Council of Scientific Industrial Research, they have a huge cohort developing. They are taking blood samples and many other samples from the normal, so-called normal individual of this employee, the permanent employee and their spouses and the retired employee. Next 5 years. So it's like framing a cohort. In India, there is no cohort. So the whatever we check, we say that this is the blood pressure, this is the cholesterol level normal. That normal cholesterol level we say based on the Caucasian population. There is a Framingham village, and from there so they started taking sample 10 years, 20 years. They initially took that service for 5 years, 10 years, 20 years, 40 years now. And from there they can tell that this is the normal value of cholesterol, normal value of LDL, normal value of HDL. We are following that village, but the culture or food or behavior is different, so we may not have the right value. So now we are making the CSI launched CSI has 37 institutes, whole India, country, Gujarat, Bengal, Odisha, every state has CSI. So there we are taking samples for next 5 years, and our markers will be checked there for the 5 years. But so we will do a multiplex ELISA, but this is the time to get the help of the engineers, electronics and others, so that we can use this in a very smart way. And also I put 6 panels because there is a scope to put AI, because there will be score. My dream is to find out the score based on these up and down regulation, a complicated mathematical equation, external equation. There you will get some score. So it's 10. 10 means very serious. 9, okay, also serious. Maybe 7, okay, fine. 5 is fine. That kind of thing will be in practice in future. We do or don't do. Caucasian will do. Americans will do, and will buy their kit if you don't know. I'm not the only smart fool. There are so many people are trying to do like this. So this is my idea. Many people are trying to address the issue in another way. So the bottom line is heart attack is predictable, and we will predict the heart attack very soon with the help of biology and the engineering. So these are my students. They are all yes, they all in Chicago University, Chicago. They was with also Philadelphia and Albert Einstein. Yeah, he's in Harvard Medical School. They did all this work and they have postdoc working there. Thank you. Also, my clinical collaborator and also other structural biologists in this work. Thank you very much.
Any questions that you can ask?
Any silly questions? No questions. Okay, thank you. Yes. Good. Yes.
Hello, sir. So, sir, how did you get the six proteins out of the 3,000?
That's the right question. Very good question. Yes, the 3,000 proteins are there. Out of that, we came out with 6 proteins. Number one, we have some hypotheses. Hypothesis is cholesterol, reverse cholesterol transport, and the heart attack, and also inflammation. So, cholesterol, hyperlipidemia, and heart attack. That is our actually story. That's why those protein match with those hypotheses, and also the inflammatory story. That's how we generate the 6 proteins. There might be other proteins, and also the intellectual ability. Many people may think other fine proteins, and these proteins are from the mass spec. There are some techniques. How is the score in the mass spec database? Score was very high, so we wanted robustness. So if you want to utilize for human application, robustness will be there. It's not a very minor change, and it is really we validated in the patient sample. It is really high changes, drastic changes. That's why we selected these proteins.
And sir, you collected the samples while heart attack was ongoing or before?
Heart attack happens. Diagnosed the heart attack. Was admitted in hospital there, and we had also normal sample and also normal 2 type of normal, absolutely normal. They don't have any problem. Which is very difficult to find nowadays. Age match control. It is very difficult to find a 50 years age person or 60 years person without any medicine. But most of the patients are 60. Most of the 50-year-old people will take some medicine, either cholesterol medicine, statin, or something hypertension and hypertension. So we found some good.
Sir, I would like to ask one question. Firstly, it was a very interesting talk. Thank you for the talk. You mentioned about one of the markers which you used was a fibrosis gene, which is PICP. So, like there are various other conditions in the body when the fibrosis gene is activated, like liver fibrosis, for example. So, like, how can you like specifically say, or how did you determine that this was the particular gene which is particularly responsible in the cardiovascular disease? Because
rheumatic heart disease.
Yeah.
That is also a very good question. You understood it. There are many occasions when fibrosis will increase, but this is fibrillar collagen 1 and 3. There are a lot of collagen. So this is 1 and 3 specific for this kind of fibrosis. There is a possibility of increasing this protein when there is a bone cancer. So you have to exclude those. Otherwise, this is specific for this hard wall fibrosis.
Following question, you said, as she also mentioned, that in the mass spectrometry, you were able to identify certain genes which were upregulated and certain proteins which were upregulated and downregulated. As we know that blood itself has a lot of proteins, a lot of various other factors which are there, it forms a corona. So, like, there can be certain proteins showing, like, mass spec basically identifies the mass of the protein, right? And based on that, it segregates the protein, and hence you can identify. So, there can be certain protein-protein interactions which are occurring, because of which mass of certain protein might be increased. Like, it's a very technical question. Like, how did you basically eliminate those false positives?
No, they are not correct. Mass is not increased. The protein is digested and then it is flight of time. So the M by Z. So interacting proteins are not, these isolated peptides. Based on that it is diagnosing, detecting. And there is a relative quantification. It is detecting and also relative quantification is there. So we have actually spiking with some known protein, not amount, MSMS. Yes, this is MSMS. So therefore it is specific and relative quantification is there. Second, even we have to validate by actual quantification method, ELISA, in the blood. So whatever we are seeing there, and also 3-peptide match, technical applicates, a lot of artifacts have to remove there and then find out this.
Okay, thank you, sir.
Sir, I just have a small question. So, sir, the 6 biomarkers that you shown, does all of them have similar procedure or parameters for sample collection?
Yeah,
All of them.
from blood.
So, sir,
From blood, all are ELISA based.
so, sir, what is stopping it from preparing a combined 6 or 4 marker based more accurate detection system?
Yeah. So that is now the business model. The scientific talk is done. Now you need a lot of money to make it 6 panel marker to multiplex or ELISA. So I'm talking to many companies and other things. This is not possible. This up to this is possible in our government setup. But we have in our government setup there are a lot of limitations. Now we have to go out of the government setup, do something with company.
But sir, it is possible, right?
It is possible, but it's very difficult to convince company that it has something. So that I am trying now. This is now business part. Next part. It's good that you understood it actually. Correct me a second.
So can lipid profile screening improve early detection of cardiovascular disease alongside of cardiac biomarkers?
Lipid profile, of course, lipid profile is a good the marker for atherosclerotic, but even the lipid profile is normal, heart attack happens. Even the lipid profile you keep normal by using a drug, atherosclerotic heart attack happens.
What is the
atherosclerotic burden you have to understand.
Sir, what is the connection of SGOT and SGPT in heart disease?
SGPT is SGOT.
Yeah.
There is a liver function test.
Okay.
May not have any connection with heart attack because if liver fails, then these are secondary issues.
Okay.
Any more questions?
Thank you very much.
As we conclude this insightful session, let us take a moment to express our gratitude to our distinguished speaker, Professor Arun, for their valuable time and knowledge. I would like to request Dr. Rahul Kumar to kindly present a token of appreciation. Thank you for that insightful session. Now, as we move forward, it's my pleasure to introduce our next distinguished speaker, Professor Mahesh Kumar. Today he will be presenting on real-time prediction model for metal oxide-based gas sensor using machine learning techniques.
Yeah, very good afternoon. So I'm going to talk about how we can use AI in the healthcare or in other sectors. So we will take one example. These are the basically metal oxide gas sensors. So these are the chemi-resistive gas sensors are there. Particularly these materials properties are changing when we expose in a particular gas. And today this gas we will talk about the H2S here. Now when this H2S emission is there, one is in the healthcare also, suppose there is a kidney disease is there, mostly we face the problem when kidney is 90% is gone. But how to do the initial basically investigations if started this one. So sulfur compound is coming. So whenever we are going in the hospitals, if you do, suppose we are going for regular check or we have some other disease, if you have some fast screening here, so from when we exhale basically, if we can get initially some idea here how much basically if some kidney related disease is there. So here, particularly in this here, we can use these sensors, and because the compound quantity is very small in the ppb level is there, so we need these sensors. They can exactly predict basically if sulfur compound is there, because same time water molecules, humidity is also there, also CO2 is there, so there are the different gases, and we want to detect particularly the H2S gas. So my research is focused mainly on the devices, materials, how to grow different kind of the materials, metal oxide, 2D materials, then 3-5 semiconductors. And by using this one, we can make the different type of the devices. Mainly here at IIT Jodhpur, our focus is mainly on the sensors. One is the radiation sensor, dosimeter is there, your alpha, beta, gamma, how we can detect photo detectors. Another one is the gas sensor, heavy metal ion sensor. This is again the upcoming, basically, particularly in our country, is the major problem now. All the rivers or water sources, basically, these are polluted. What we do in the industry, for example, lead, mercury, we are using for the different applications. In the textile, we use lead-based dye, and now the water we release without the basically purifying this one, because the overall cleaning process is there, that cost is very high, and generally the industries basically they save the money and they release almost in Ganga river, Yamuna river, now heavy metal ion sensors here, their level is increasing. Again, here we have started work on AI and ML, how we can use this one. So particularly, I will talk today about the H2S. When food decompose, H2S emission is there, even in the kitchen, septic tank, and daily in the newspaper here we see this one, casualty happened because of H2S, ammonia, and methane. These are the three particular gas, but particular here the majority is from the H2S here. So we can use basically these sensors for the different applications. Only the concentration difference is there, because when we exhale, basically kidney problem is that H2S quantity is in ppb level, otherwise here level is in ppm here. So we need here sensors for the different applications here. Also in the oil industry, because coal, basically same procedure is there. There also particularly this H2S gas is there. So if you design any type of the sensors, these are the basic characteristics. Here, whatever quantity we are measuring, there should be a high sensing response. That means whatever electrical or quantity we are measuring, With respect to input, your change should be high, response should be fast here. Again, the power consumption should be less. So if you are making the biosensor, if you are making the chemical sensor, any type of the sensor, these are the basic characteristics to make basically these sensors. So to make these gas sensors, we can use different type of the sensor. Even we can use the gas chromatography, but gas chromatography cost is 15 to 20 lakh is there. We can get the exactly, suppose your vehicle pollution, or even if you want to use for different here, we cannot spend this one. So we have to make the basically low cost, and we can use here the conductivity based here. So if you take some material, and these materials basically property is changing when we are exposing in a particular gas here. So in this case, suppose if you are exposing in H2S, your conductivity is increasing or decreasing. That is depending on the here what type of your oxidation or reduction is happening. So particularly we will focus on this approach here, chemi-resistive approach, and then how we can make basically these highly reliable sensors. So first here, chemi-resistive sensor was invented in 1960 by the Japanese scientist Taguchi, and he used the metal oxide, zinc oxide. So particularly we take two electrode, and in between electrode here some resistance is there. So now how particularly this resistance is changing when we are exposing in the different gases. Again, to improve, because here gas sensors, your reaction is taking place at the higher temperature. If bulk is there, we go 200, 300. And if you can make the nano, we can reduce the operating temperature 150 or 100 degree. And we can make the micro heater, this one, by using the MEMS technology. So we can reduce. But still they have basically these sensors, they have the drawbacks. One is the here low sensitivity and selectivity. So suppose we are making for H2S, but because of the humidity or oxygen, again we get the interference particularly, and the sensitive to environmental factors. Suppose you are in Chennai, Ahmedabad or Jodhpur, humidity level is different. So our sensors should not be affected by the environmental factors. And power consumption, these are the drawback here. So can we overcome basically these drawbacks here by using the AI and machine learning here? So we can combine, one is our sensing for here, sensors, and then we can use basically these algorithms. So how these chemi-resistive sensors are working, this is the, we can understand to explain this one simple concept. I think you have heard about this materials, semiconductor materials. There are two types, one is P type and another one is the N type. How diode is working basically. So in one direction your current is very high, in opposite direction current is almost negligible. This will allow only in one direction. So in the P-type semiconductor, Fermi level is close to valence band. In N-type semiconductor, Fermi level is the close to the conduction band is there. So Fermi level is deciding basically your semiconductor is intrinsic, N-type, P-type. That means your carrier concentration is what type and what is the level is there here. Now if we join this one here, what will happen? In P-type, we have holes are majority carriers, here electrons are majority. In N-type, when we join both these semiconductors, N-type and P-type, your carriers will start diffusing. Because here we have concentration, hole concentration is high, and in this case, electron concentration is high. So electrons will start diffusing from N to P, here P to N, and they are leaving the ions. These ions at the interface, particularly, these are not mobile ions. Basically, there is a difference. Electron and hole, these are the free carriers. They can move basically by applying the potential or by basically if carrier concentration gradient is there, they can move from higher concentration to lower concentration. So what will happen after some time, one depletion region will fall, and here electric field is developing. That is from positive to negative. Now the further diffusion of holes from P to N and electrons from N to P, this will stop. So there are two forces are working. One is electric field, another one is the diffusion. And in the steady state, what we will get? We get the depletion region, because free carriers are depleted from this region here. So we call this is the depletion layer is there, depleted layer is there. Now further diffusion only possible if you are applying the bias. So in the diode here, we are taking one electrode from here, another here. So if you connect P type, we will get the high current. If P type, if you connect with the positive, if you connect with the negative, all concentration will decrease. So almost this will stop the your current in opposite direction. This is the simple, your PN junction is there. You have studies basically in the 12th or your first year basically how diodes are working. So same concept we can use basically to understand how these gas sensors, chemiresistive gas sensors are working. Now if I take basically, suppose this is the N-type semiconductor is there, and if you take one contact from top and one we take from the bottom electrode, basically if we deposit the metals, what will be the resistance of this material? By Ohm's law we can calculate R equal to rho upon L. So depending how much width is there and what is the cross section area is there, by the Ohm's law we can calculate here. Now resistivity in the semiconductor depending on the doping, but we are not changing the doping, okay? Whatever doping is there, we are taking this one. So here resistance will be the constant. If you are not doing anything, basically if you are not changing the dimension, your resistance, whatever this is 5 kilo or this one 10 mega ohm is there, whatever range is there, this will be the constant. Now, if I bring, hypothetically, if you think basically, if you bring near to this one P-type semiconductor, what will be the impact on the resistance? Suppose I am bringing P-type semiconductor near to this one. Here, your electrons and diffusion will start. When diffusion will start at the junction, we will get basically depletion region. Now, what is the impact of this depletion layer on the resistance? So carriers are depleted from this region. Now we don't have free electrons, particularly in this region. Depletion region is depending on the doping concentration. So what will happen? Your cross-section area is decreased. Now cross-section area is here A minus NA is there. So if cross-section area is decreased, your resistance will increase. So same if suppose gas molecule is coming near to this surface, if gas molecule is taking the electron, what will happen? Here depletion region will form, and depending on the how many number of molecules are coming. Suppose we are taking the different concentration of P-type semiconductor, this depletion layer will be small or this one will be high. Accordingly, our resistance will change. So this material here properties, basically your resistance is depending how many number of molecules we have for a particular gas. Suppose this molecule is taking the electron, here this your resistance change in the resistance will be high. If you have only less molecule, change will be small. So we can relate basically, we can relate here particularly this resistance with the concentration, and then we can make the different type of the gas sensors. Only here criteria is there, this metal here your semiconductor should absorb or basically release the electron here. Some interaction should happen this one between the particular material, then we can make this type of the sensor, this can be resistive based gas sensors. Then for again to understand this one, suppose this, there are different types. One is the direct reaction is there, another one is the indirect is there. So in this case, here suppose this is the nanowire is there. Initially oxygen is also taking, because in the environment we have the oxygen everywhere. In some materials, oxygen is taking electron from the semiconductor and is making the depletion region. Now the channel width is there, this is small, and suppose I bring hydrogen near to this one, here hydrogen and oxygen will react and depletion width will change. So now the conduction will be high, is depending on the here radius of the channel here. So we have to take these basic concepts to make the different type of the gas sensors. One, directly your gas is reacting, another one is that with oxygen is reacting, and then basically we are getting the change in the resistance. Here this we can take the electrode, these materials property, basically their resistance is changing when we are exposing in a particular gas. And now if we remove the gas, our sensor should come basically in its initial stage. This is the conventional approach is there basically to make these chemi-resistive sensors. When we expose, their resistance is changing, increasing or decreasing depending on the nature of the gas. And then when we are removing the gas, again our sensor is coming in initial stage. But here we are facing some problem. Here what is happening, when is the baseline stability is there. When we are removing the gas, sensor is not coming in the initial condition. And another is here basically non-linear responses are there. Again signal fluctuating because of the humidity and other gases. So these are the basic problems, but we can overcome these problems by using the machine learning approach here. That is not possible by in the conventional method, because from last 50 years basically we are focusing to remove how baseline stability we can get, because sensor if is not reliable, we cannot use at least for the healthcare. Okay, we need the very high accuracy, and here issue is that baseline stability is the issue is there. So in the new approach here we can come, we are using basically new approach where we are measuring the impedance and applying the machine learning. By using this one, exactly we can predict the basically how much percentage of a particular H2S gas is there. So here, and then we can use for the different applications here depending on the basically requirement. So these materials, again N-type or P-type is there. In the P-type, accumulation layer is forming, here depletion layer is forming depending on the type of the gas, which type of the gas we are using. So in the conventional approach, what we are doing this one, we are applying, we are measuring the resistance. How we measure the resistance? We apply a constant voltage, we measure the current here, and then we can calculate what is the resistance of this particular device is there. Now, depending on here from one to another, how particular this gas is moving. So here one is the depletion layer is forming, and another one is the your between electrode and material is the junction is there. We call the basically here your electrode contact is there. These are the interconnects is there. Depending overall resistance, your variance is depending on these two factors. And what we are doing in the conventional approach, we are taking only resistance. Okay, we are applying the DC voltage, measuring the DC current, and then here we are considering only resistance. But another part is also here, because when depletion region is forming, your capacitance is also there. So we can measure the impedance, where we can measure basically capacitance. Also we can measure the resistance, and then we can apply the machine learning. Then accurately we can predict these cases, and we can use for the healthcare applications. How we are measuring the resistance? Just as I said, this one we can apply the constant voltage, measure the current, and from V equal to IR we can calculate here. But if you are measuring the impedance in your capacitance or inductance is there, one more term is involved, that is the phase is there. So here, how much change is there in the phase when we are exposing the gas or when we are removing the gas? Phase also we can measure, because for machine learning AI you need a lot of data. We can use all these data, then we can put in the algorithm, and finally we can get output. Otherwise we have only one data, and that data is noisy. Our output is there, that also will be noisy. But if you have more data, we can develop these algorithms and we can predict accurately. So we can use basically here, we don't have the inductance, particularly we have the capacitance. So at different frequencies we can measure. Otherwise in the DC there is no frequency impedance. In a range of frequency we can measure this data. So we can get more data as compared to your DC resistance is there. So here in the direct current, we apply the voltage, and this one measure the current, and we can calculate the basically resistance. Similarly, if resistance we have, by AC also we can calculate, because both are on the resistance, both are in the same phase, and we can calculate basically how much resistance is there. But if we have inductance or capacitance, there is a phase is there. So in the presence of the gas and in the absence, if there is any change in the phase, basically that also we can relate with the gas concentration, how phase is related with the gas concentration here. So now we can measure the more data, particularly when we are measuring the depletion region capacitance here. So these are the basic concept is there, how we can measure the impedance, how impedance is different from the resistance. That is also the basically if you are doing at lower frequency, your resistance and impedance both are same, but higher frequency, your capacitance is that it will start playing the role here. So what we are doing here, we are developing the different materials. This is one example. If you want to develop a H2S sensor, we can deposit on the silicon SNO2 material. We can make this IDE here by using lithography or by using the shadow mask. We can decorate with some materials to improve their sensitivity. We do by the sputtering. I will not go in the detail. These are the deposition parameter. So just for you, this one we are depositing some material, and these materials are absorbing the H2S gas. So this is the final device is there, how this look like. We do the material characterizations, ground material is same. So here is the SNO, this one SNO2, and this is the copper oxide. By electron microscopy we see the image, because here reaction is the chemical reaction is there. So if you have nanomaterials, more surface area, we are expecting the change in the response will be higher as compared to the bulk. Next, what we do this one, simply we measure their resistance. When we are exposing in 100 ppm, 50 ppm, their resistance is the function of carrier concentration is there, as we discuss in the mechanism. So now how much percentage is that we can calculate. So we are measuring the change in the resistance, this initial resistance, how much when 100 ppm or 2 ppm, 5 ppm we are exposing. By decorating this one copper oxide, particularly their response is increased. Initially is 60 to we can reach around 70, but more than this one is there is the selectivity is there. So here in the controlled way, we are decorating one more material that is giving the higher this one sensitivity and selectivity. Here you can see this gas is not selective, because we are getting response from methane, ammonia, other gases. So now how to identify particularly only H2S emission is there, because sensor is the blind is there. So one way is that to improve this one, we can decorate with different basically your catalyst, and then we can make selective towards a particular gas. So this is the first way where we can decorate the different your metal particles or another materials as a catalyst, and then we can improve their selectivity and sensitivity. So we made this one selective by decorating with the 60 seconds. When we are going higher, this one again this started the decreasing. Now the question comes, if copper oxide is giving H2S, why we are not using the copper oxide? This one we can deposit only copper oxide and we can make. But what is happening? Copper oxide, when reacting with the sulfur, is making the copper sulfide, and copper sulfide is the highly conducting is there. We need the semiconducting to make chemiresistive sensors, not the conducting. Why? Not the highly conducting. Relative change, because we are measuring the relative change, how much relative change is there. That should be the significant. Okay. If relative change is not significant, in that case we cannot measure basically. So in the conductors, change is almost negligible is there, because conductors they have the your carrier concentration is very high. If you add in that 1000, your electrons will be the same. Okay, so because of this one, we have to use the semiconductor material, so we can increase basically the change is the significant is there. Again, we can understand, for example, Ambani will give you 5 crore. Will his lifestyle will change? but your will change definitely. Okay, so same way is that if rich person is donating small amount, but if someone who is having small amount, suppose I will give you even 10 lakh rupees also, definitely that impact will be on me this one. So depending on this one, because conductors they have large number of carriers, so if you are adding or taking, their number is not changing. In semiconductors is less number of electrons. So if you are adding or taking, basically there is a large change is there. So we are measuring the change in particular here. Next, what we have done, we have looked for the another material same way as we do on the devices. I can skip this part, basically is similar, only we have changed the material, but you can see the change we are getting higher as compared to the previous material. And again, here you can see this is highly selective and other materials response is minimum when we are decorating with the PD metals. So first at the laboratory level, we optimize on the silicon wafer with the existing technology, and then we make the prototype sensor. So here we take the alumina tube, we deposit by the sputtering this material on the alumina. You can see here is the two contacts, and this is the heater is there, and particularly we operate at 150 degrees centigrade. By IR camera we can measure how much temperature is there, and this is the simple sensing setup is there. Is this cost is less than the 50,000 is there, only we are using this airtight basically boxes. Here is our sensor, this is the gas inlet is there, and by this digital board we are measuring how much particular impedance is there. So in this board we can vary the frequency from up to the 1 megahertz at different frequency, and then we can measure the impedance. So this is the schematic diagram is there. We have the H2S cylinder, from PPP to PPM we can go, and then we have basically air is there. We are using the air. So we are mixing with air, and then we are finding out how sensor is responding. These are the data at different frequency, how impedance is changing. This is the real part of impedance is having two parts. One is real part, another one is the imaginary part, and third is coming the phase. So how phase is basically changing. And this is the between real part and imaginary part. You can see with different ppm here, particularly how this basically your impedance data is varying. So what we have to do here, we can get, so lower frequency, this is the 100 hertz is there. So we can consider as a this one as a only resistant. So here when we are going one cycle to another cycle, here data is not stable. That as your baseline is not stable, we can consider this, we are measuring only the resistance. But when we are going at the higher frequency, at this particular frequency around 10 kilohertz, we are getting the stable data. Again here the phase is giving more accurate data. So how phase is changing? So when gas molecules are coming, basically the phase is also changing. So we can measure, do all these measurements, impedance, real part, imaginary part, total impedance, phase at different frequencies, all data we can here now how to do the basically analysis part. So we can use this machine learning algorithm where we can put your ANN or CNN model is there. All these data we can put in the model, and now we know particularly how much our concentration is there. So we can initial data we can use for the training purpose, then testing purpose, and then a known value we can predict. So we have used here ANN model is there. We have optimized these parameters basically by using the different optimizer. Here these how much should be the epochs, error function. These are the important, because when we are this one epochs are higher, basically we get the more accurate value, but still your this time is increasing, your calculation time is increasing. So we have to optimize all these parameters here. So here you can see in the pink color, this is the actual data is there, and here we have taken the individual data and then we are close to basically predicted value is there. This is the predicted value, here is the actual value. So our model is that NN model is giving accurate value basically almost close to the our actual value. So we can use this AI to basically predict the accurate concentration and then we can improve their performance. So here we have used single frequency and then we have done so, but if we see there is a difference between the initial data and this difference is there because sensor is taking some time, that is the from sensor side because your temperature stabilization is taking place. So if you are doing on a single frequency, this is the issue, but at the higher, this one, in the higher cycle, we are getting close to the actual values. So next we have used the multi-frequency model and then we have also introduced the humidity. So if you are doing this one zero humidity, 20%, 40% here. So zero and this 0 and 20% data we have used for the training and testing purpose, and here we have predicted and we are getting almost equal close to the our actual values. These are again we have done lot of this one, use the this your NN model and then we have found, and another parameter is the selectivity. So we have mixed the with H2S with NO2, ammonia, CO2, hydrogen, methane, and then we found this one we are getting almost the signal we are getting that we are getting only from H2S here because we have used the higher concentration. H2S is 10 ppm and these other gases we have used the higher concentration. So using this one we have given 10, so almost equal to 10 ppm from our model also we are getting this data. Now we can use, again, if you want high accuracy for the healthcare, we should not this one trust on one sensor, we can use the sensor array. So we have made the sensor array, for example, 6 sensors. So if from one sensor or 2 sensors, if we are getting the noisy data, that we can ignore by using these algorithms. So you can see these 4 sensors is working. One is for humidity, another one is particularly what is the oxygen level is there. All we can integrate on a single chip and then we can make. Here you can see basically when we are going at lower, because from the when we exhale basically kidney failure is there. This H2S emission is there. That one is in the ppb level is there. So here 500 ppb or below is there. So we can use this array and then accurately basically we can measure from humidity because at different humidity slightly data is affected. So from the machine learning we can eliminate the humidity effect and we can accurately measure. So here you can see basically how sensor is responding. We can use here copper oxide and when copper sulfide is forming. So from this one we can get this one also. We can use the heater side also. We can use the pulse modulation and we can reduce the here operating power also. So here you can see this one ppm or the other gases we have taken the higher and the response is almost negligible is there. So we can also reduce the power if you are using the continuous heating or pulse heating. Here we can use the different model and we can save the power. Also from the array we can get the accurate values. So this work is done by my PhD student Amit and postdoc Bharat. He is helping in the machine learning algorithms, is developing whatever from the sensor we are getting the data, and then we have the actual data. So we can use these different basically sensor data and ML model to get the accurate values. So with this one, thank you, and if you have any questions.
Hello, sir. Thank you so much for such insightful talk. So I would like to ask that mostly metal oxide based sensor face the temperature problem, higher temperature problem. And since you have made the heterojunction of CuO and SnO2, so still the temperature is 150 and you came down to 100 degrees Celsius. But whenever we talk about the power consumption or the real-time monitoring, it should work on room temperature, or maybe some factor can specifically include into the lowering, more lowering the temperature. So what would be the possible solutions to break down this heterojunction into the room temperature conditions?
So one thing is that we can use because we have to give the energy that reaction should take place basically. Your charge transfer should take place this one.
Yeah.
So we can apply in the form of basically photo activation.
Yeah.
That is the another way is that basically by we can use because now gallium nitride technology is matured everywhere we are using the LED. So we can use the UV LED because we have to give the energy, otherwise your reaction will not take place. Here we are giving the thermal activation, so we can give the photo activation. Another we can develop the material, 2D materials basically. They are giving the response at room temperature, but recovery is not complete. So we can do some basically from the data side. Once response is coming, small response basically, then we can switch on the heater for a shorter time. Then we can get we can overcome this power consumption problem.
Right. And sir, one more question. So basically nowadays researchers are targeting the flexible sensor and the wearable part of these sensors. So rather than making a proper device fabrication or the complex fabrication, if a person is rather than wearing a sensor itself and it can directly connect it to Bluetooth and anything. So what is the material point of view for the flexible sensing part so that they can directly be incorporated onto the wearable substrates and then can be connected into a Bluetooth devices or some kind of form?
Now, if you see all smartwatches, they are using the basically your electronics part.
True.
That is not flexible.
Yeah, that's
Yeah,
not possible.
but watch is flexible. The same way, basically, we can reduce their size and then we can make. Or we can use basically on the flexible substrate, that is the other possibility.
Okay. Thank you so much.
Any more questions? I think everyone is hungry. Yeah.
Nice lecture. So when you showed that sensor is detecting other gases, carbon dioxide, other than sulfur. But the value for sulfur is very high, other is very small.
Yeah.
So you can reach a threshold and cut off,
Yes.
and then you can get a sensitivity.
So that we need the algorithm basically. And also same time we are planning to connect with ammonia and methane because these are the three gases basically.
Okay.
Okay. So then exactly from ammonia sensor how much data we are getting from methane how much we are getting, and then we can basically use the machine algorithm whenever this level is below and how much exactly is there. So yes, we have to detect from
Because the actual the sulfur value is very high.
yes.
H2 value is very high.
Yes. So that is from the sensor side.
Sensor side.
But still small response is there from other cases. Now we can use the ammonia sensor, we can use this sensor and then we can detect the data from overall. So that we can do from the EM.
Right. Thank you.
Thank you.
Thank you for that insightful session. As we conclude, let us take a moment to express our gratitude to our distinguished speaker for their valuable time and knowledge. I would like to request Dr. Nishan Sharma to kindly facilitate Professor Mahesh Kumar.
participant can be taken one group photo. Come.
अरे बंद कर दे लाइव स्ट्रीम बंद कर अरे बहुत चोट है बहुत मतलब आखा दिवस में हाँ स्टॉप स्ट्रीम